Hello, everyone, and welcome back for more biotechnology. It is time for us to start a new chapter, and if you've been keeping up, our focus lately has been on protein production and protein purification. So now seems like a good time to take a step back and start looking at kind of where it all started for us. We're going to start talking about biotechnologies involving DNA. So let's go ahead and get into this. So, this is going to be the start of rehashing several discussions that we've already had. So, number one, something that should make sense by now, all cells contain DNA, right? So, human cells, plant cells, E. coli cells, like the one you see depicted in the bottom right here, they all contain DNA molecules. Now, we've talked before about the sorts of characteristics that separate prokaryotic cells like E. coli and other bacteria from eukaryotic cells like plants and animals, in that the DNA is stored a little bit differently. But the basic rules behind how DNA operates and how DNA stores genetic information is going to be the same from cell to cell to cell. So at any given time, DNA inside of a cell, so if you look at the E. coli cell here, we have the chromosomal DNA down here, and then small pieces of plasmid DNA here. We've seen both of them throughout the semester. DNA at any given time is either being replicated, so we're either making copies of it by using the enzyme DNA polymerase, or alternatively, what we do a lot more frequently is we take small segments of the DNA, which of course we call genes, and we transcribe them into messenger RNA by using the enzyme RNA polymerase. And that is all for the purpose of protein synthesis, as we have seen before. So, what we've basically described here is the whole central dogma. DNA gets replicated, DNA gets transcribed into RNA, and RNA gets translated into protein. That is basically how a cell operates on a minute-by-minute basis. That's basically all it does. So, having a good understanding of the central dogma and previous more involved discussions on the central dogma that we've had before, basically all detailing how proteins get made, that's going to be really essential to understanding a lot of what we're going to be focusing on in this chapter. How do we manipulate DNA for our benefit, whether it be for developing therapies for diseases, developing things that, technologies that are less expensive than what is already out there. We've seen examples of that with chymosin and insulin and things of that nature. So, before we really start talking about technologies involving the manipulation of DNA, you want to make sure that you've got the central dogma down at this point. So let's talk about chromosome number first. So all organisms have a particular number of chromosomes carried by each cell. Now this number is not necessarily unique to the organism, but every cell of that organism should contain the same number of chromosomes, as long as those cells are not sex cells. So to this end, organisms usually are either going to be haploid or diploid. Haploid organisms carry only one copy of each chromosome, whereas diploid organisms, like us, like humans, we carry two copies of each chromosome per cell. So if you look at the diagram on the right, in which all 23 types of chromosomes are lined up head to tail, you'll notice that we have two copies of chromosome number 1, two copies of chromosome number 2, and so on and so forth until you get to the sex chromosomes, the X and the Y chromosome. You'll notice here that this individual must be male because this person has an X and a Y chromosome. A female would have two X chromosomes. So for us, for humans, because there are 23 different human chromosomes and we carry a total of 46 per cell, that must mean that we get 23 of our 46 chromosomes from mom and 23 of them from dad. If you end up taking a more involved biology class or some sort of developmental biology class, you'll learn about the process of meiosis and why it is that this ends up being the case. So a fairly useful technique called a karyotype allows for the isolation of the chromosomes out of cells, and we can actually stain them and visualize them and then kind of piece them together like a puzzle so that we can check to make sure that all the what we call homologous pairs of chromosomes match up and everything looks on the up and up. So the main reason to do a karyotype is when usually a baby is born and you want to check to make sure that there are no gross genetic abnormalities, such as big segments of chromosomes being deleted, having too many or too few chromosomes. So usually you're looking for gross abnormalities like that. Okay, so a lot of what we're going to be focusing on in this chapter is DNA replication. So this is going to basically form the backbone of a lot of the technologies that we use that involve the manipulation of DNA. So we've tackled this sort of thing before. All we really said about it back then was that DNA replication is what we call semi-conservative. What that means, if you need a little bit of a refresher on the subject, is that if you have a double-stranded DNA molecule, and if you peel that DNA molecule apart, each one of those single strands can act as a template for the synthesis of a new complementary strand. So it's semi-conservative in the sense that we are not just synthesizing a brand new double-stranded DNA molecule from scratch. We're taking the two pieces of the original template DNA and we're making a new single strand for each. It's semi-conservative to indicate that we do conserve the two original strands of DNA, but we're not just making it from scratch either. So in vivo DNA replication, the phrase in vivo refers to processes that happen naturally inside the cell. So anytime you see the word in vivo, that means this is how it happens in nature. This is how the cell does it. So the actual in vivo process of DNA replication ends up being fairly complicated, right? So it's kind of a jumbled mess. There's kind of a lot going on. So what has to happen for DNA replication to occur in vivo is that you don't just need DNA polymerase, the enzyme that actually synthesizes the new DNA strand. You need a slew of other enzymes as well just to get the following things to occur. So number one, you need to unzip the double-stranded DNA. You have to peel the two strands apart from each other so that each single strand that is no longer bound by the other strand can serve as a template. This unzipping is promoted by an enzyme called DNA helicase. So once you have the double-stranded DNA unzipped and you have exposed single-stranded DNA, unfortunately, DNA polymerase doesn't know what to do with that. So DNA polymerase can't just bind to a single strand of DNA and make a complementary strand to pair with it. So DNA polymerase needs double-stranded DNA. It needs at least a small segment of double-stranded DNA. So another enzyme called RNA primase is actually going to bind to the single-stranded DNA and synthesize a very small RNA primer that is complementary to the single-stranded DNA. That way, once DNA polymerase comes in, it can recognize this now double-stranded segment of DNA, and it can extend that RNA primer and use that as the way to start the DNA synthesis process. So one thing that you will notice as you look at the picture here is that because we do not just completely unwind the DNA and separate it into two different segments, we actually unwind the DNA kind of like a zipper, which is why we used the term unzipping before. So because we're unwinding the DNA bit by bit, as we do this, there can be a lot of rotational torsion and tension built up in the DNA strand, and that can be kind of a problem. So the way to kind of think about it is twisting a rubber band. So if you twist a rubber band, you can really get to the point where there's so much tension in the rubber band, if you let go, it snaps back and it rotates pretty violently. So another enzyme called DNA topoisomerase is necessary to relieve the building tension in the DNA strand as new DNA molecules are built. And then finally, the last enzyme that is necessary, we've talked before about the differences between DNA and RNA, so we really don't want to have DNA that has little bits of RNA in it, which we still do at this point because of the actions of RNA primase. So another enzyme called RNase H is going to come in and cut out those RNA primers so that they can be replaced with the corresponding DNA nucleotides. We don't want any uracil or any of the RNA nucleotides in there. We want to replace those with actual DNA. Okay, so at that point you should probably be convinced that, okay, DNA synthesis in vivo, pretty complicated, right? Well, it's a little bit more complicated than that. So part of the issue is that we've discussed that DNA is always synthesized in one particular direction, from 5' to 3'. This means that if you have an incoming nucleotide that is going to be added to the growing DNA polymer, the 5' phosphate of that nucleotide is going to be attached to the 3' hydroxyl group of the nucleotide that the strand is growing from. So this means that DNA is always going 5' to 3', 5' to 3'. You cannot synthesize DNA backwards. You cannot go from 3' to 5'. So for one of the strands, so keep in mind looking at the picture, because both strands are being used as a template, we are going to make the best use of this by building new DNA molecules off of both of them. So the issue here is that because we are using both strands as templates, one of the synthesis reactions is going to be a little bit awkward. So the two strands that are serving as our template here are called the leading strand and the lagging strand. Things are pretty simple actually for the leading strand. Because of the way that it is being unwound, DNA can be continuously synthesized from 5' to 3', and it doesn't cause any problems. But for the lagging strand, in order for DNA to be synthesized 5' to 3', eventually the synthesis is going to hit a roadblock, which is what we call the replication fork. The replication fork is the point at which the DNA helicase has basically the point at which DNA helicase has currently unwound the DNA molecule. So if you look up here towards the top, this is what we call the replication fork. So DNA polymerase on the lagging strand is synthesizing DNA towards the replication fork. So DNA synthesis can't happen any faster than DNA helicase can unwind. So this is going to cause a little bit of problem. This is going to cause what's called discontinuous DNA synthesis of what are called Okazaki fragments. So what we're dealing with here is the fact that because DNA polymerase is going to run into the replication fork, we can only synthesize these little fragments of complementary DNA one at a time. So what that means is that as the lagging strand is being worked on, we're only going to synthesize bits of complementary DNA, and there's going to be gaps in between them. So those are going to have to be filled in later. So your big takeaway from the past two slides should be this. In vivo DNA replication is very, very, very complicated, and it seems like it's the sort of thing that is going to be very difficult to replicate in vitro. In vitro referring to things that happen in the laboratory. So if we're going to get DNA replication to happen inside of a test tube, we need to find a way to simplify it, because this seems like an absolute nightmare to try to get to happen outside of the cell. So the good news for us is that DNA synthesis in a laboratory setting is much, much, much less complicated, because really the only enzyme that is going to be required here is DNA polymerase itself. We don't need helicase, we don't need primase, we don't need topoisomerase, we don't need RNase H. All we should need is DNA polymerase and a few other things. Those other things are going to include the template DNA, of course. You can't get away with not having that. So you need a very, very small amount of the DNA that you wish to be replicated. That makes sense. You do obviously need DNA polymerase. There's no getting around this. So this is the enzyme that catalyzes the addition of new nucleotides as we build a complementary strand on top of the template strand. You do need DNTPs, which are the deoxynucleotides, the adenine, the thymine, the cytosine, and the guanine. These are going to be used as the building blocks for making new DNA. They are the substrate for DNA polymerase. So if DNA polymerase is the enzyme that builds the new DNA molecule, it's going to need the building blocks in order to do that. We will need primers. We will need both forward and reverse primers, which are just short pieces of DNA oligonucleotides that are going to anneal to both the 5' ends of both of our DNA strands that we want to replicate. So even though things are going to be a bit simpler here, we still cannot get around the requirement that DNA polymerase has in that it cannot work on purely single-stranded DNA. It's got to have duplexed double-stranded DNA in order for it to work. And then finally, because this is all happening in a test tube, we will need some type of reaction buffer that is buffered to maintain a pH of about 8.0, which is what DNA polymerase seems to like. And then other necessary reagents and cofactors like dithiotherotral, which helps to keep a reducing environment that DNA polymerase likes, magnesium, which helps the DNA polymerase to work on the incoming nucleotides, and then other various salts that are going to be necessary as well. So if you look at this list of reagents that are required for in vitro synthesis, it looks a lot better than what we were looking at before. We don't need much else other than this. Okay, so that is going to do it for this introductory video into DNA biotechnology. So join us next time and we will start delving into specific applications before we really start hammering at the major topic of this chapter, which is polymerase chain reaction. That'll probably be the third video in the series. So thanks for your attention, and I will see you next time. Bye-bye.